Hi guys, let's dive right into section 2, the replication of DNA. DNA replication is the process of copying a DNA molecule. It is semi-conservative, meaning each strand of the original DNA helix serves as a template for a new daughter strand. So each, quote, new strand of DNA actually contains an old strand. Only one side is new. Okay, one side is the original template. So here we have our parent strand. It opens up, okay? This is called the replication fork because it's like a fork in the road. And one side is used as a template to build a new strand of DNA. Okay, and the same thing is happening over here. Okay, now remember that our DNA is anti-parallel, meaning that one side is facing forward, the other side is facing backwards. So you'll notice that one side is just a continuous strand that is following the replication fork. So as this moves, as it opens wider, the DNA strand on this side will just keep getting made and keep getting made. But over here, okay, it's working in the opposite direction. So it starts here and it moves over here, but eventually that replication fork is going to close. So these are called Okazaki fragments. And after they are made, an enzyme will come through and bond all of them together. But I'm getting ahead of myself. We'll get to that in a little bit. The process of replication requires the following steps. The unwinding or separation of the two strands of the parental DNA molecule by an enzyme called DNA helicase. Okay, it's called that because it's breaking the helix of the DNA. It also requires a single-stranded binding protein called SSB to attach to the newly separated DNA molecule to prevent that helix from reforming because it really wants to bond. It wants to stay together. And unless it has that protein there, it's going to reattach. Okay, it's going to stick back together and you're not going to be able to copy it. DNA replication also requires that complementary base pairing happens between the new nucleotide and the nucleotide of the template strand. An enzyme called DNA primase places a short primer on the strands of the DNA to be replicated. Okay, remember, primer is like what you put on the walls before you paint. Okay, it's preparing it, it's getting it ready for the process. And then another enzyme called polymerase recognizes that polymer. Okay, it's made out of RNA. It's like primer is not exactly the same thing as paint. RNA isn't exactly the same as DNA, but they're similar. So the polymerase sees the RNA there, and it's like, oh, guys, we got to start. So it attaches to that polymer, and it begins to bring in new nucleotides based on what the parent strand of DNA has. So if it has an A, it's going to bring in a T. If it has a G, it's going to bring in a C. So this is the process of joining nucleotides on one strand. It's continuous. Okay, that is the leading strand. The lagging strand makes little bitty chunks called Okazaki fragments, and an enzyme called DNA ligase will attach them together. Each daughter DNA molecule contains one old strand of DNA and one new strand of DNA. That's why it's called semi-conservative, because some of it's conserved, some of it's the old stuff, but not all of it. Replication of DNA in prokaryotic cells. Bacteria only have one single circular loop of DNA rather than the chromosomes that we have. So replication moves around in a circular moves around the circular DNA molecule in both directions, and it produces two identical circles, two identical pieces of DNA. The process begins at the origin of replication, and it takes about 40 minutes, but the cell divides every 20. So a new round of replication can begin before the previous round is completed. So basically, it's always copying, it's always getting copied, and it's always getting ready to divide. That's how prokaryotic cells replicate so quickly. That's how bacteria grows so fast. Eukaryotic replication. It happens at numerous points on the chromosome. The DNA unwinds and unzips into 2 strands, and each of those strands serves as a template for the new strand. Complementary base pairing forms a new strand paired with each old strand, and this is done by DNA polymerase. A lot of times it's abbreviated DNA pol, so capital D, capital N, capital A, and then lowercase p-o-l. Replication bubbles spread out bidirectionally until they meet. So, like we said on the previous slide, replication starts at a bunch of different places on a chromosome, and it spreads out until they all meet. The complementary nucleotides are joined. So the chunks from each replication bubble are bonded together to form one continuous strand. Remember, the replication is semi-conservative, so each new strand has 1/2 new and 1/2 old DNA. Polymerase is unable to replicate the ends of chromosomes, which are called telomeres or telomeres. Both pronunciations are acceptable. Telomeres are copied by an enzyme called telomerase, and unregulated telomerase activity negatively affects the cell's function. So if the telomerase is replicating too much, if it's adding too much of a tail, it's bad for the cell's function. If it's not adding enough, that's bad for the cell's function. Here is a picture of prokaryotic and eukaryotic replication. So you'll notice here we have our original prokaryotic strand. Okay, so it starts replication at the origin, and it starts forming a new strand here and a new strand here. It grows larger and larger until we get 2 complete strands of DNA, 2 complete circles. The blue strand is the original, and the green strand is the new. Now you'll notice in eukaryotic cells, we start replication in multiple spots. These are the replication bubbles, and they spread out further and further until they meet. Where they meet, they will be bonded together by ligase to make 2 complete connected strands, and it will look something like this. Once again, the old strand, the parental strand, is blue, and the new strand, the daughter strand, is green. So here is a diagram showing all of the different enzymes that are required for DNA replication. So we have DNA helicase here that is separating the helix. We have our binding proteins that are keeping the helix from sticking back together. Okay, so that's keeping the replication bubble, that replication fork, open. We have DNA polymerase that is copying. It's bringing in those new nucleotides. Okay, so this is our leading strand that's copying continuously. Remember, it goes 5' to 3', but this strand is not in the proper orientation. So we have another DNA polymerase that is finding that primer that was laid down by DNA primase, and it's attaching to that, and it's copying until it hits this second primer here, and then it stops. And another enzyme is going to stick those fragments, those Okazaki fragments made by the DNA polymerase on the lagging strand. It's going to bond them together. So we have a DNA ligase, and it's taking the end of one strand and attaching it to the end of the other strand. So I know that that's a lot. We're going to spend some more time talking about this, so don't freak out. If you have any questions, let me know. The replication of DNA is very accurate. A DNA polymerase is very accurate, yet it makes a mistake about once per 100,000 base pairs. But the cell is capable of recognizing those errors and correcting them. So the DNA won't bond back together correctly. It'll be like, oh, hey guys, we got a problem here. This isn't right. And it will go back and fix it. So that's what I got for you guys. If you have any questions, let me know.